Okay, hello there. This is Rohan Mishram again. In your previous lecture, what we have discussed was what are the application of these bioinformatics and chemoinformatics approaches in drug discovery, and we have came to conclusion that these computational method are used for minimization of efforts that are usually required for drug target identification. You can use this bioinformatics and chemoinformatics method for validation of the identified drug target. It can also be used in identification of suitable compounds that can be used as the further hits. You can use these computational approaches in lead identification as well as in lead optimization. So to continue with, I would first like to give you a brief idea on how things are going to work in our course of syllabus. Right. So this slide represent you a general protocol for identifying a suitable drug candidate or performing computer aided drug discovery project, a small computer aided drug discovery project, if you wish to implement it in your own project also. Okay, so in this slide you can see that there are two sections, right? So there would be you can say this procedure can be classified of having multiple aspects. So the first aspect of your protocol would be identifying the drug target, and the next aspect would be identifying or collecting the compound library. It would be related to compound collection and processing, and the first aspect, as you can see over here, would be related to identifying a drug target first. Okay, so let us begin with, I would just let you know we are going to deal with each and every aspect in more details of whatever has been shown on this slide. Right. So the very first step would be identification of drug target. Then we would seek out if whatever drug target you have identified, well, if it contains an experimentally solved structure, we would check out in the database called as PDB. So this PDB is a database where people deposit, or scientific community deposit, the experimentally solved structure of proteins and other macromolecules that can be relevant for therapeutic interventions. Okay, then in PDB we ask ourselves a question: if the structure of the identified drug target, if it is experimentally solved, or if the structure of the identified target is present in PDB or not. Of course, you would have two ways over here. It is entirely possible that the structure has been solved. If that is the case, you further proceed to identify the active site of the drug target, or you can say you are supposed to identify a place in your drug target, a protein, where your compound or whatever compound that you have designed might go and attach. That place is called as active site, or we can say that place is called as binding site. So where exactly your compound would go and attach, we would term it as active site. Okay, active site has got some technical definition, but for time being, we would consider as active site or binding site in analogous manner. Okay, so once you have identified an active site in your drug target, we would call it now as a target protein. Next, you need to process that target protein, and you would finally get your target protein ready that can be used for further procedure. Okay, well, now let us go back to the question, where you can find a negative answer for the question that is the structure available. So what if the structure of your identified target protein is not available in PDB? In such case, you can develop the structure of the same identified target using a computational technique called as homology modeling. So this homology modeling is supposed to be used as a computational technique in the scenario when you don't have experimentally solved structure of your drug target. Okay, so by using this method, you would end up with a three-dimensional structure of that protein, and once you have got the structure, again you would go on for identifying where exactly your compound would go and bind to that protein in terms of its active site or binding site, and proceed in the same way as we have proceeded with the experimental structure. Okay, so the objective of this step is to identify a suitable drug target and prepare it for further processing. Okay, now another aspect of the procedure would be to create compound library. Okay, so from where you would collect the compound, how would you filter it, how would you generate the 3D coordinates of those compound. Okay, and what are the different filters that are applied to it. We are going to deal with all these aspect of compound library preparation in this portion. Okay, so to begin with, well, let us first discuss this lecture. We would we would you know we would devote to drug target identification, on how do we identify suitable drug target. Okay, so before we begin, well, I would just give you an example on let us first try to identify, or let us try to first understand what exactly is a drug target. Okay, so this drug target can be any macromolecule, usually it is protein. Okay, now these proteins can be, well, say these proteins can be either enzymes, or they can be ion channels, or they can be transporters, or they can be receptors. Okay, so these proteins can either be enzyme. Enzyme means it would perform certain enzymatic activity, it would catalyze certain biochemical reaction, right? What are ion channels? Ion channels are proteins that are responsible for maintaining the ionic balance across the membranes. Those are included in ion channels. On the other hand, receptors are supposed to be present on the cell surface or membranes of your cells, wherein the role of the receptor would be to get some signal from external environment and pass that signal inside the cell, so that cell can behave in a precise way in order to sustain the stimulus that is present outside of the cell. Okay, so usually in therapeutic context, these three type of protein macromolecule are used as a drug target, right? So there are a lot of enzymes that are used as a drug target. Well, say for example, I would cite an example of cyclooxygenases, which we call it as COX. Well, these are example of an effective anti-inflammatory drug targets. Okay, so whatever anti-inflammatory drugs that are present in market, that might include your aspirin, diclofenac, ibuprofen. Well, all these small molecule would go and bind to enzyme called as COX-2 or COX-1. There are two isoforms of COX. Okay, so after binding to this COX enzyme, they would inhibit its activity, and that would result in some therapeutic effect, like you would deal with inflammation, right? These COX inhibitor are usually used as painkillers, right? You know that diclofenac is prescribed as one of the painkiller. Ibuprofen is prescribed as one of the painkiller. So is the case of nimesulide, so is the case of aspirin, so on and so forth. There are many anti-inflammatory drugs. In a similar way, in case of receptors, there are G proteins that can be used as receptors. Most of the anti-cancer drugs target these G protein coupled receptors. Okay, so these receptors have got multi-domain structure. It has got one domain that is present on the outside of the cell membrane, where your ligand usually or your signaling molecule usually goes and attach, and on the other side of the membrane you have got tyrosine kinase domain, and this tyrosine kinase domain is usually responsible for further signaling process. Okay, so in this way these proteins can act as drug targets. They can be in form of any enzyme. The drug targets can be in form of ion channels, they can be in form of receptor, a G protein receptor, or some sort of other receptor. So by inhibiting these proteins, usually some therapeutic effect is carried on. Okay, now, well, let us try to identify the process by which the drug targets are identified. How do we end up like, what proteins from a pathogen can be used as a effective drug target? Okay, so of course there are two methods by which one can identify the drug target. You can say that one can identify the drug target using experimental approach. Another is something called as the theoretical or computational approach, which is now being very popular for identifying newer drug targets. Okay, so I would just let you know first how experimental methods are used for identifying the drug target. So the very first method is called as phenotypic screening. So what do we do in phenotypic screening is that we screen the compounds in cellular or animal disease model, and then the objective is to identify if our given compound is responsible for causing desirable changes in a phenotype. Well, say for example, for this I would like to cite the example of, say, cataract. Okay, so in sugar induced cataract, what we do is that we have a cellular model. Well, say for example, we can isolate the lens of the animal and induce cataract it in. Okay, and when in that lens, in that cultured lens, when we induce the cataract, then we can test our own compound and check out if our compound would result in decreasing the cloudiness of the lens or not. So in this way what we are trying to do is that if we give our compound to any cellular, or you can say animal disease model, do it brings out the desirable effect. In our case, in the example that I have cited, the desirable effect would be the cloudiness of your lens should decrease when you give your compound to that cataract induced lens, right? This is called as phenotype. Okay, initially it was cloudy, but after giving your compound, it has it changed its phenotype to again transparent. So in this way you can figure out if your compound is able to characterize, or if your compound is able to bring out the desired chemical, or you can say phenotypic effect. Okay, phenotype is something that you observe. Okay, another example that I would cite is in case of your anti-inflammatory response. Okay, what we do in mice model is that we have a technique called as carryogen induced paw edema. What we do is that we take an animal model like mice or rat, and we inject it with carryogen, and carryogen we know is responsible for bringing out inflammation. So the paw of that mice, we observe that it become inflamed, उसमें सूजन आ जाती है, right? And what we do is that then we next give that mice model or animal model with our compound, and then we test, or then we observe if the inflammation on the paw of that mice decreases or not with time. If the inflammation decreases, that means अगर वो माइस का पॉ के ऊपर में अगर वो inflammation कम हो जाता है, that means our drugs or whatever compound that we are testing work as an anti-inflammatory agent. Well, that is how you try to figure out if your compound is working or not by something called as the phenotypic screening. Okay, next we have something called as gene association study. So what we do in gene association study, I would like to cite example of genotyping of BRCA1 or BRCA2. So these two genes are used to determine susceptibility to breast and ovarian cancer. Okay, so if an individual is associated with certain type of genotype, then if there is mutation in such genotypes, then such individuals are more susceptible to breast and ovarian cancer. Okay, uh, what you can next do is that you can rely on biomarkers. Okay, so biomarkers are nothing but what I can say is that in certain disease condition, we'll say for example in case of cancer, you would observe that there is some elevated expression level of circulating tumor markers like HER2, HER2. If there is a lot of expression of HER2 gene, that would mean that there is something wrong, and it act as a biomarker that the person is suffering from cancer, and thus it can be used as drug targets as such. Okay, so these were a few of the experimental techniques by which one can identify if your compound is working right or not. Okay, then the next type of approach that is used for drug target identification are included in computational approach, right? So the very first approach that we are going to discuss, a very first technique that we are going to discuss for identification of drug target is called as comparative metabolomics, or sometimes it is also referred to as subtractive genomic approach, right? The next approach that we are going to study in more detail is something called as pharmacophore probing, or something called as pharmacophore mapping. How this pharmacophore mapping can be used for identifying a drug target, we would deal these two topics in more details. Okay, so first let us begin discussing this comparative metabolomics approach for drug target identification, or what we call it as subtractive genomic approach for drug target identification. Sometimes people use these two terms interchangeably: comparative metabolomics or subtractive genomics. Okay, so, well, let us begin with how can we identify a drug target using comparative metabolomic or subtractive genomic approach. To understand this concept, first you need to understand what exactly is metabolome. Okay, so well, let us first define this term. What is metabolome? So you might have heard bioinformatician or these computational biologist people use this word called as genome. How do we define the genome? Genome is usually defined as the entire genomic content of that organism, of any organism. We would refer it to as genome. In similar way, people have also coined the term like proteome. Proteome correspond to the entire protein content of a cell, and hence we say that entire protein content of a cell is called as proteome, and the study of the entire protein content of a cell is called as proteomics. The study of entire genomic content of the cell is called as genomic. Similarly, we have identified the term called as metabolome. That is, by this term, we would refer to entire metabolic content of an organism. So, whatever metabolic pathways that are present in that organism, we would call it as its metabolome. So metabolome would you can consider metabolome as entire metabolic content of an organism, a collection, or you can say the collective of all of the metabolic pathways that are present in an organism would constitute the metabolome of that organism. Okay, I would just give you an example. What you see over here represent the entire metabolic content of an organism. Each and every small this represent the metabolic pathways that are present in the organism, and as you can see, everything is related to everyone. So just to give you a brief idea, I would now show you the simplified view of metabolism. This is what these computational biology people usually use to intimidate the non-biologist guys who have just came into biochemistry or computational biology. Okay, so I would just like to give you an idea on the view of the entire metabolic, how the metabolism is organized in cells as such. Okay, so right from your 12th standard, you might have I would zoom into this so that you can check out in more detail. So the very first metabolic pathway that you might have studied right from your 12th involves your glucose metabolism. As you can see, the glucose usually the glucose usually begins with the glucose metabolism usually begins with glucose being converted to glucose 6 phosphate, then fructose 6 phosphate, fructose 1,6 bis phosphate, and hence we call this pathway as glycolysis, that would end up into creation of pyruvate, and this pyruvate then further enters into something called as TCA cycle, or what we call it as Krebs cycle. Okay, and from this Krebs cycle, whatever the NADH or the reducing power so created, it enters into your electron transport chain and eventually result in formation of ATP. Okay, so usually this is taught first because somehow if you see the entire metabolic network, it appears to be central in position, and every other metabolic pathway is somehow related to this central metabolic pathway. Okay, I would just give you an idea, like all of the glucose metabolism, whether it is in form of polysaccharides, or whether it is in form of hexose, or whether it is in form of this pentose, all the sugar metabolism over here is shown in this green color. Okay, the photosynthetic reactions are shown over here. All the lipid metabolism, that is fatty acid degradation and fatty acid formation, whether it is in form of biosynthesis or degradation or a phospholipid metabolism, that is shown over here in blue color. उसके बाद में over here you have isoprenoid metabolism, that is shown in brown color. So is this porphyrin metabolism, that is also shown in brown color. Steroid metabolism is shown in blue color over here, right? On the other side of the map, you can see the amino acid metabolism, like aromatic amino acid metabolism. Aromatic amino acids that usually include your indole containing tryptophan, tyrosine. Well, that is shown in red color. Okay, catecholamine metabolism is again shown in your brown color. Purines and pyrimidines nucleotide metabolism over here is shown in this uh, what is this color, blue shade of color. Okay, and again other remaining amino acid metabolism is shown in red color over here. Okay, so in this way, the objective of showing you this map is that for you to appreciate that metabolism is highly complex network of metabolic reactions that are connected to each other. Each and every metabolic pathway, each and every biochemical reaction is somehow connected to other metabolic reaction or other metabolic pathway, and this gives an enormous complexity of the biochemical reactions that do happen in a cell. Okay, so my ultimate objective of showing you this poster of metabolism is for you to appreciate that this metabolism is quite complex. It is quite dependent on each other. Everything is related to everything else in the cell. Okay, so what I mean to say over here is that metabolome constitute all the biochemical reaction that are present in that organism. Okay, that is called as metabolism. And the study of such metabolome is called as metabolomics. So metabolome means what? It would constitute the entire metabolic content of an organism. That it is. Okay, I hope you are now aware of this term. Okay, so now what we do is that in comparative metabolomics approach, what we do is that we would compare this metabolic network of one organism with another organism. Okay, so the objective of doing such a comparison is that we would try to compare the entire metabolic content of two organisms and try to identify which are the metabolic reaction that are present in both the organism, which are the set of metabolic reaction that are present in only pathogen, and what are the set of metabolic reaction that are only present in human. Okay, well, say for example, we would take an example of any pathogen. We'll say for example, it can be either plasmodium that is responsible for causing malaria, or you can say another pathogen you can consider is leishmania, which is known causative agent of a disease called as leishmaniasis. So what if you want to find out some drug target using this comparative metabolomics approach for any pathogen like plasmodium or leishmania? So if you want to find out such drug targets, what you would do is that you would compare the metabolome of these two organisms. In our case, we would consider the pathogen plasmodium or leishmania, and we would compare its entire metabolome with that of human. The objective is to compare the two network of the metabolic pathways. Okay, so the main concept behind this approach is that these two organisms has evolved differentially. That means it is entirely possible that in due course of evolution, this Plasmodium or Leishmania might have evolved certain metabolic pathways that are not present in human, or on contrary, it is also possible that humans might have evolved some metabolic pathways that are not present in this pathogen. Our objective of comparing these two metabolism is to identify certain set of reaction that are uniquely present in that pathogen which are not present in humans. Okay, that is the ultimate objective of comparing these two metabolomes as such. Okay, so when you compare two metabolomes, you might want to classify the reactions into three major categories. The first category would correspond to all of those reactions that are only present in human. Okay, another category would be all of the reaction that are only present in pathogen. Okay, and third category might be there might be certain reaction that are present in both the organism. Okay, so of these three classes of reactions, you would focus on the set of reaction that are present only in pathogen. We would call such set of reaction as unique reactions that are present in pathogen. Okay, so now once we identify such unique reaction to pathogen, well, if you have identified a reaction, that means you have identified an enzyme, because most of the reaction in cells are catalyzed by enzyme catalyzed reaction. Okay, are usually those enzyme catalyzed reaction. So you have identified a reaction, that would mean you have identified an enzyme, and therefore such enzyme that catalyze a unique reaction are usually a very good drug target. Why? Because if you stop the activity of such enzyme, you would stop functioning of that reaction, you would eventually stop functioning of that metabolic pathway, and in this way you would create disturbances in that pathogen, and eventually that would result in some therapeutic effect. Okay, so the ultimate objective of doing this metabolic comparison is to identify a reaction and eventually identifying an enzyme that is responsible for that unique reaction, and hence they can act as an efficient drug target. So enzymes of such unique reaction form a very attractive drug target. Why? Because that enzyme is uniquely present in that pathogen. If you stop the functioning of that enzyme, it would only hamper the pathogen, leaving the host uh, host intact. Okay, that won't interfere with human. In our case, the host is human. It won't interfere with human, and it would specifically go and target to the pathogen itself. Okay, so well, uniqueness is one of the most important property that enzyme should possess in order to be used as a drug target. Okay, so besides uniqueness, whatever enzyme that you have identified, if that enzyme is catalyzing something called as choke point reaction, it would be more advantageous. Okay, so whatever unique and choke point reaction, now say for example you have identified, we are going to talk into more details of what is choke point reaction. uh, First, you would need to identify the enzyme that are unique. Then whatever unique enzymes that you have identified, you need to make sure that it would be more advantageous if that unique enzyme do catalyze the choke point reaction. Then you would make sure that whatever enzyme that you are supposed to target, that are unique, that are choke point reaction. They have some vital or essential function in pathogen of sorry, in physiology of that pathogen. Okay, and final criteria would be that these enzymes should not have any close homologue into human. I'm going to deal with each and every property in more detail. Okay, well, so for example, first let us discuss what is choke point reaction. So choke point reaction are such reaction in a metabolic network that either uniquely consumes a specific substrate or it is responsible for producing a unique product in a metabolic network. That is how we define a choke point reaction. Okay, so now enzymes that are responsible for carrying out such choke point reaction are called as choke point enzymes. Okay, to give you a visual aspect of this concept, over here you can see I have given you an example of choke point enzymes over here. Okay, so the enzyme that are or the reaction that are shown in a red arrow correspond to your choke point reaction. As you can see over here, this enzyme or this reaction is responsible for uniquely consuming a substrate. Okay, over here you can see this reaction is responsible for creating a unique product. Okay, in this case you can see that this particular reaction consumes this metabolite that is the product of multiple reaction. Okay, over here this enzyme which has got an EC number 1.3.99.5, this enzyme kind of consumes this specific substrate that is product of multiple enzyme or multiple reaction. That is, multiple reaction would converge to produce a single metabolite which is consumed by this particular enzyme. On the other hand, in this metabolic network, you can see that this particular reaction is responsible for creating this metabolite which forms a substrate for multiple reaction which is present on the other side, or you can say it forms the source of a metabolite that give rise to multiple reactions, multiple metabolic reaction or multiple metabolic pathway eventually. So what would happen is that inactivation of this enzyme or inactivation of this enzyme would eventually result in disruption of that metabolic network. So now let us consider what would happen to the metabolic network if you stop this reaction. If you stop this reaction, eventually all of the metabolites that are present downstream of this reaction would accumulate, right? And that would eventually result in toxic accumulation of a lot of metabolic metabolites as such. Okay, on the other hand, if you stop such reaction that produce a unique product, if you stop this reaction, all the subsequent reaction or the subsequent metabolic pathways that are dependent on this metabolite would be stopped. Eventually it would result in starvation, right? All the metabolic reaction would stop. That would result in starvation of certain metabolite. Okay, so in this way, thereby a choke point enzyme served as a very potential drug candidate. Why? Because if you stop a single biochemical reaction, it would hamper the entire metabolic network. Okay, and if such a metabolic you can say if such a choke point enzyme is unique that would create a very severe effect in that pathogen, right? So this is how whatever unique reaction that you have identified, that would also be advantageous if it is a choke point enzyme. Okay, so whatever enzyme you have identified, it should be unique, it should follow choke point enzyme. Okay, then your effective drug target should be vital. Okay, so what our objective is that whatever enzyme that we have identified, it should go and stop certain biochemical events. Okay, so now there are, you can say, compensatory pathways that might result in formation of the same metabolite. Okay, I would, in other words, I can say that any of the biological system, well, it can be in form of pathogen, or it can be in case of your human, they have some compensatory metabolic pathways, right? Well, say for example, to cite an example, uh, well, say for example, uh, in your purine biosynthesis, or you can say nucleotide biosynthesis, there are two pathways by which these nucleotides are produced. The very first pathway is called as de novo pathway, wherein your nucleotides are produced step by step from scratch. That is what we call it as a de novo. And there is another pathway called as salvage pathway. Okay, in this salvage pathway, what we do is that your cell would create this metabolite from whatever that has been left. That is called as salvage pathway. Okay, so now one might think ki, okay, these nucleotides are very essential component for DNA metabolism, and what if I stop the synthesis, or what we say de novo synthesis of this metabolic pathway called as nucleotide metabolism में अगर मैंने de novo pathway अगर stop कर दिया, तो one might think कि if I stop that de novo pathway, there would be no nucleotide. If there are no nucleotide, then there would be no DNA, and if there is no DNA, that pathogen would die. So in this way, one might think that stopping de novo pathway might act as a very good therapeutic strategy. So if we stop this de novo pathway, we can kill that pathogenic organism. But this de novo pathway has got some compensatory pathway called as salvage pathway. So even though if you target enzymes from this de novo pathway, cell or that pathogenic organism would use then salvage pathway to create the same metabolite, and it would have no effect. Okay, so in this way, by the process or by the term vitality, we mean that we should target such an enzyme that holds a very key position in that pathogen physiology, that if you target that enzyme, if you stop that enzyme, its inactivation would eventually lead to the death of that parasite. And if such enzymes are targeted, that would result in pathogen fatality. Okay, so vitality is, you can say, the concept in which you make sure that if that particular enzyme, which is very vital for that organism survival, is to be targeted. Okay, therefore, the drug target that you are supposed to identify, it should be vital in the pathogen physiology. There should not be any compensatory pathways that would result in creating the metabolites that are, created by those drug targets as such that you are going to identify. Okay, so that is called as vitality. In short, vitality is nothing but a property of an enzyme such that if that enzyme is deactivated or inhibited, there should be no alternative way for that pathogen to produce those metabolites that are created by that vital enzyme. That is what we term it as vitality. Okay. The next criteria is that we'll say, for example, you have now identified enzymes which are unique. You have identified the unique enzyme that follows the choke point reaction. You have now made sure that whatever choke point enzyme that were unique are very vital. If you uh, if you if you destroy that enzyme, the pathogen would eventually die. That would mean that the enzyme is very vital. And after conducting all these tests, you would have to make sure that the human genome or human, or you can say host, should not contain any close homologue to the identified drug target. Okay, so what are homologues now? Homologues are enzymes or protein which has got similar function. Okay, it should not happen कि you have identified a drug target in your pathogen whose similar enzyme is present in human. So your drug target should not go and bind to any of the enzyme which is similar to that of the one which is present in your pathogen. Okay, it should not happen that you have identified a drug target and similar enzyme is present in humans. So it is entirely possible that your drug target would go and curtail both of the enzymes. So that should not happen. Okay, so inferring non-homology, that would assure that any homologue of identified drug target protein in host should not be curtailed by a drug identified to inhibit the pathogen enzyme. That is what I want to tell you in short. ओके, तो कोई भी ड्रग टारगेट आपने आइडेंटिफाई किया तो थेरेटिकली उसमें 4 प्रॉपर्टीज रहनी चाहिए. पहली प्रॉपर्टी ये कि दैट शुड बी यूनिक टू योर पैथोजन. यूनिक है इसका मतलब क्या कि अगर वो एंजाइम को आपने इनिबिट किया तो फर्क सिर्फ पैथोजन पर पड़ेगा, फर्क आपके होस्ट के ऊपर में नहीं पड़ेगा. सेकेंड वन, whatever unique enzyme that you have identified, that should be choke point reaction. अगर choke point reaction अगर होगी तो वो enzyme को अगर आपने inhibit किया, that would eventually result in either accumulation of the metabolites that would result in toxicity, or it can result in starvation of metabolite जिसकी वजह से वो पैथोजन मारा जाएगा. Third property should be that it should be vital. वाइटलिटी अगर एंजाइम में रही तो इसका मतलब क्या कि अगर वो एंजाइम को आपने इनहिबिट कर दिया, the pathogen would eventually die. And the final criteria should be that the enzyme should be unique, or you can say उसका कोई भी क्लोज रिलेटिव आपके ह्यूमन में नहीं रहना चाहिए. Why? Because enzymes tend to have similar structure, and your drug should be able to identify कि okay, this is where I am supposed to bind and this is where I am not supposed to bind. Okay, if similar proteins are present in both the enzyme, sorry, if the similar protein are present in both the organism, your drug would go and inhibit both the enzyme, and eventually it would cause off target toxicities in case of host. ओके, इसका एग्जांपल मैं देना चाहूँगा कि if you try to target any enzyme say for example, um, say for example, if you try to target you know certain enzyme like dihydrofolate reductase, or say for example, if I try to target any enzyme which is present in pathogen, and if its related enzyme is present in human, then since the enzyme structures are very similar, your drug would go and attach to both the target enzymes. Okay, so it would create problem in such a case. Okay, so now summary of target identification would be comparative metabolomics. It would be used to identify unique enzyme. Then you would shortlist the enzymes which are unique, those follow the choke point reaction. Then again you shortlist the choke point enzyme that are vital. Then again you shortlist the enzyme that are not homologous to host. And finally you would get the final target enzyme. So what we are doing over here is that maybe you would start out with comparing entire metabolic content, in which you would compare a few thousand reaction, those would be catalyzed by few thousand maybe enzymes, right? Then you would shortlist the enzymes that are unique. तो ये 1000 में से हो सकता है कि there are only 100 enzymes that are unique to only pathogen. तो हो सकता है कि ये 100 enzyme में से only there might be some 70 unique enzyme that follow this choke point reaction. हो सकता है कि ये 70 choke point में से एंजाइम में से there are only 30 reaction that are very vital. तो हो सकता है कि ये 30 में से there are only 5 enzymes that are not homologous to human, and in this way you would end up with only 5 enzymes that can act as a final target or final drug target as such. Okay, so in this way you begin with say some 1000 enzymes that you have compared. You have shortlisted only 100, those are unique. Then again you have shortlisted say only 70 that are choke point reaction. Then again you have shortlisted only 30 that are vital, and of those 30 only 5 are you can say that are not present in human, or whose such enzymes that has not that don't has got any close relative to human being. So in this way, in each step you go on reducing the number of enzymes, and hence we call such strategy as subtractive genomic strategy. At each and every stage you go on reducing the number of the enzyme. Okay, so such an approach is called as subtractive genomic approach, or such an approach is also sometimes referred to as the reductionist approach. Okay, and in this way you end up with something called as final set of enzyme that can now be further used as drug targets. Okay, so well, I would just list out the softwares that can be used in such an study. So the very first software that can be used for comparative metabolomics is an online server called as Rahanuma. Rahanuma is an online tool wherein you need to identify the two organism, also for example human and pathogen, and this software would then compare their metabolic pathways and let you know that okay, of the two compared metabolomes, we have got say 100 to 250 or whatever reactions that are uniquely present in pathogen that are not present in human. So you can use this tool for comparing the two metabolome. Okay, so there is another software or online tool called as Met This Explore is used for identifying the choke point reactions. Okay, so it performs this choke point analysis. Then vitality is confirmed by comparing, or you can say a blast based search in a database called as database of essential genes, what we call it as DEG. So this database consists of genes or a number of genes that are known to be highly important, or you can say that database corresponds to a set of genes that are known to be very essential for survival of life. So now whatever enzyme that you have identified, if you compare or if you try to figure out if such enzyme is present in that database, and if you find out कि whatever choke point enzyme that you have now identified उसका sequence वहाँ पर already present है, that means that enzyme is vital, and thus you are able to identify if that enzyme is vital or not. Okay, so how would you now make sure that जो कुछ भी एंजाइम दैट इज वाइटल ना उसका कोई भी क्लोज रिलेटिव आपके ह्यूमन में नहीं है. So what you can do is that you can compare whatever vital enzyme that you have identified to उसका सीक्वेंस आप कंपेयर करेंगे आपके होस्ट के साथ में, दैट इज ह्यूमंस के साथ में. So if you end up using PsyBlast after various iteration of PsyBlast, if you still don't get any hit, then you can be of surety कि human में भी इसका identical क्या close relative भी present नहीं है, and such enzyme might be used as final drug targets. Okay, so in this way, we have today discussed subtractive genomics approach or reductionist approach or comparative metabolomic approach for identifying the drug target. Okay, I think this much would be enough. In our next lecture, we would deal with more aspect of this metabolic simulation that can be implemented for identifying a drug target. Okay, so it is enough for now. I think we should stop.